All right, now moving on to the next section in chapter 15. We're going to cover 15.8 and 15.9. So now we're going to be looking at certain cis factors, cis-acting elements, and trans-acting elements that work on the regulation. So cis-acting elements are just regulatory sequences, and they're sequences, therefore they're part of the actual DNA, right? So they are things like promoters or other types of elements that we'll talk about, like enhancers or silencers, that basically have an effect on the transcription. Now, these cis-acting elements don't actually do anything by themselves. They're just regulatory sequences that other trans-acting elements, so things that are not DNA, like proteins, so transcription factors, will come in and bind to these specific regulatory sites in order to actually, put on and pass on the different various regulatory mechanisms. So cis-acting DNA sequences can regulate transcription as well, but again, not by themselves. They have to have these like regulatory proteins in regions where these proteins can act to stimulate or repress transcription. All right, so one of those first cis-acting elements are, of course, promoters. And we know that promoters have a very specific position. They're always found upstream of our structural gene, right? Or our gene, actually, in this case, we're talking about, eukaryotes. So any gene, every single gene has a promoter for it. So promoters are basically about 100 nucleotides long. It's a nucleotide sequence that serves as recognition sites for transcription machinery. So it's located immediately adjacent to genes they regulate, so immediately upstream of them, and they specify where transcription begins and the direction of the transcription. Okay, so whether the transcription goes forwards or backwards, right? Because until now, I mean, it's very easy to show the transcription going towards the right, but it can go towards the left depending on where the promoter is and what it actually encodes. So they have things known as promoter elements, so specific sequences, consensus sequences as well, that basically a lot of eukaryotes have that don't really change. These sequences don't really change because transcription factors or other regulatory factors bind to these specific sequences. And so if these sequences change too much, then of course the regulatory factors are not going to bind to them, and therefore we may have all sorts of horrible consequences that can arise. So these promoter elements, we'll look at them in a little bit of detail in like the next or 2 slides or something like that. But this includes things like the TATA box, the CAT box, GC boxes, right? The TATA box is a box that we find about minus 30 nucleotides away from the first, about approximately 30 nucleotides away from that first transcription start site. It's very similar to the PrivNow box that we know very well in bacteria, right? So it kind of, this is where we have a lot of opening up of our DNA, and the DNA is now single-stranded, and we can actually start transcribing a little bit from that juncture. So the importance of specific promoter sequences is evident by mutational analysis. So again, as it's everything with genetics, in order to study these things, we had to mutate them, and we looked at what particular phenotype or characteristic came about from these mutations, and then deduced essentially why these promoters or these sequences were important in the first place. So there are 2 kinds of promoters, 2 subcategories in the eukaryotic promoters. We have core promoters and then also proximal promoter elements. So core promoters, these are the ones that are usually like the normal ones, hence the word core, that are RNA polymerase II. If you remember our RNA polymerase II, this is like the main one that basically transcribes the majority of all mRNA and even some tRNA and RNA. Then we have things like RNase polymerase I, which basically transcribes rRNA and tRNA from their encoding genes. So core promoters are determine the accurate initiation of transcription by RNA polymerase II. So they're found essentially in front of most of the genes that will then become proteins, most of the genes that have to become mRNA. We also have proximal promoter elements. These guys are present and they modulate the efficiency of basal level transcription. So those genes that have to be constitutively expressed, so continuously expressed, those housekeeping genes, usually under the control of proximal promoter elements, because they always need to have at least some level, like some basal to minimum level of transcription. Core promoters can also be 2 different kinds. We can have focused or dispersed promoters. Focused are, they're the ones that specify the transcription initiation at a single specific nucleotide, which is called the transcription start site, that +1 that I always write. It's a major type of initiation for lower eukaryotes, but not as many vertebrate genes are found under the control of the focus promoter, about 30% of them. Usually it's associated with genes that are very highly regulated, that have a lot of different components to their regulation. And a lot more is known about focus promoters than their counter other ones, the other type of core promoters, the dispersed ones. So dispersed promoters, this too should be in bold, FYI. Dispersed promoters, these guys are not found really too close to the genes that they regulate. They direct initiation from a ways away from their actual, the transcription start site. So they could be located over 50 to 100 nucleotide regions, like it's over 50 to 100 nucleotides away from the transcription start site. So it's still relatively close, but not necessarily like right next to it, like with the focused one. Not as much is known about the dispersed promoters compared to the focus promoters, the core promoter, focus promoters. But they do contain a couple of elements that make them, that are similar to the focus promoters, though the sequences aren't exactly perfect. But they essentially, they direct transcription from a number of weak transcription sites located over 50 to 100 nucleotide regions. So they're found pretty much not right next to them. So focus promoters have a couple of really important DNA elements or DNA sequence. Of these DNA sequence, just something to keep in mind, not every single one is going to be discovered in one promoter. A promoter usually has one or more, but they're not universal, right? So they're not universally found in all focus promoters. One of them is the INR element or the inner element. This encompasses the transcription site. We also have the TATA box. The TATA box is a really important one. Like I said, it's very similar to the PRIVNOW box. It's quite essential. A lot of basically any gene that is made into mRNA has the TATA box because RNA polymerase II binds to the specific region, and it's about 30 nucleotides away from the transcription site. We also have BRE, that the BRE element is usually found either upstream or downstream of the TATA box. Then other things like MTE and DPE, which can be Hold on, sorry guys. Sorry about that, guys. The UPS man came, and so that happened. Sorry. So back to the different elements that we find in focus promoters. We also have things like MTE and DPE. These are found downstream of the transcription start site. So some of them also have elements. So some of these focus promoters also have elements that you normally find in proximal promoter elements. So remember, proximal promoter elements or proximal promoters are, they modulate the efficiency of basal level transcription. So they're basically always active, but at a very minimum amount. Well, core promoters, you turn them on or off. And so you can still find a couple of these elements in proximal promoters. So yeah, these elements act along with the core promoter elements to increase levels of basal transcription. So this includes a CAAT box. Also, it can also be the CCAAT box, and then the GC box, which is just a myriad of guanines and cytosines. So all of these basically work together. So not all of them will be found in one single promoter, but each promoter has a variety of different arrangements and assortments of these particular elements. And if you see here, this is a graph that tells you the relative transcription level with this particular promoter that has the GC box, a CAT, and the TATA box. And this, essentially all of them work together to give you the initiation of transcription. So promoter elements are essential for basal or low levels of transcription. But then we have other DNA elements that really kind of amp things up, either amp things up or actually even completely, inhibit and really, what's the word I'm looking for? Really shut down the actual gene. So these more dramatic changes are basically carried out by elements known as enhancers or silencers. So enhancers and silencers, the thing about these guys is that they are not necessarily found immediately next to or adjacent to our gene. So even with distal promoters or other proximal promoters or dispersed promoters, they're found relatively near essentially the gene, right? And they're right next to it. These enhancers and silencers don't have to do that. In fact, they can be found upstream of the gene. They can be found within the gene. They can be found downstream. They can be found a ways away. So their position isn't very big. And so this is one of the main things that really distinguishes enhancers and silencers from promoters. Another thing is that enhancers and silencers don't really interact with RNA polymerase. This is not what RNA polymerase needs to bind to in order to actually start transcription. So enhancers and silencers just simply increase, or as silencers, as you can imagine, repress the level of transcription. So enhancers are modular and contain several short DNA sequences, increasing transcription rate. They can be located on either side of the gene, some distance from the gene or within the gene. They're necessary for achieving, they're necessary for achieving maximum level of transcription. So again, remember, a lot of promoters, they're there for just low levels of constant transcription. Enhancers just amp things up. And a lot of times, enhancers are very tissue specific or time specific. So a specific tissue that say is your, I don't know, your muscle tissue, okay, will have specific muscle, they're not called muscle enhancers, but enhancers that will make those cells specifically muscles, right? Muscle cells. So they have, all of them have in common that particular enhancer. Silencers are basically the opposite. So again, they're cis-acting elements just because they are DNA sequences, but instead of activating or amping up the level of transcription, they're repressing it. So they're also responsible for time and tissue-specific gene expression, but this time they're just making sure that anything that should not be transcribed or active in that cell or at that specific development time is not being transcribed. Okay, so you can see here how the enhancers could be a ways away from the actual promoter and the actual gene. Okay, these are just different kinds of examples. So insulin, we have the tissue-specific enhancer that makes it, particular to the pancreas, like islets, the Langerhans islets, and then the enhancer, right? So all these different things. In addition, we also have transcription factors. So these are going to be your trans activators, right? Or your trans working elements. So transcription factors are essentially proteins, and they have DNA binding domains that will specifically bind to those cis-acting sites like the promoters, the enhancers, and the silencers. So they target the cis-acting sites and then of the genes regulating the expression. They're quite simple. Activators are going to increase the level of transcription initiation. Repressors are going to decrease and repress the levels of transcription initiation. Some of them are expressed in a very tissue-specific way, and therefore they can regulate their target gene for tissue-specific levels of expression. Some are expressed in time-specific ways or due to an external signal as well, so that external signal will basically activate them, whether they're the activator or the repressor, and then they pass on that signal and what's supposed to happen due to that signal onto the genes. Some may be continuously expressed. However, they may only be active if modulated by phosphorylation or co-activator binding. So the phosphorylation will probably actually activate either the activator or the repressor, and then they can carry out their job. And then a co-activator is usually something that then goes ahead and helps the activator actually, increase the level of transcription. These are all just extra little levels of regulation. This is what we'll find a lot with eukaryotes. There's so many different levels of regulation, and they just fine-tune the actual timing and the amounts, right, the concentration of how much the transcription is initiated, how much transcript we're going to actually make. And this is a particular example of a gene in a human. As you can see, we have all sorts of different kinds of elements involved, right? All sorts of core promoter elements. I don't expect you to remember this. Is just an example to tell you how intricate even just one gene can be. And all of this is necessary to allow just for the transcription of that one single gene. You can see a few trans-acting factors as well, like the glucocorticoid receptor or the AP2 or even the receptor PZ120. All these things will just basically have, or even like the transcription factor 2D, MTF1, all sorts of things that basically work together with all of these cis-acting elements, which are found directly on the DNA, to basically transcribe this one single gene. So that's it for this section, and I'll talk to you guys in the next, in the upcoming video.